I would like to talk to you today about basically my research for the last year or so, where I try to ask basically what we can learn about gene regulation, or how we can view gene regulation through the lens of a deep learning model. So in some sense, understand the implicit theory of gene regulation that a deep learning model has learned in that way, and to understand how it makes its predictions. So the basic background here is that recently in the field, we have seen sort of two kinds of explosions. First of all, there's been an explosion of data that studies gene regulation. So there's been these huge projects like ENCODE, like Phantom 5, who have collected a lot of different experimental data sets that measure different aspects of gene regulation, particularly of transcriptional regulation. So we have data of transcription factor binding in many different cells. We have data of DNA accessibility in many different cells. And most importantly for the talk today, we also have data on gene expression, or more correctly would be transcript abundance as measured by cage sequencing in many different cells as well. And the second explosion we have seen is the use of deep learning models to train on and ultimately predict these sort of data from sequence directly. And illustrative of this, and probably currently the state-of-the-art model in the field, is this Enformer model made by DeepMind, which takes a very large amount of sequence. So from the center, there's 100 kilobases in every direction. It then feeds the sequence through a lot of deep learning layers, and ultimately it predicts basically the entirety of ENCODE. So all these different tracks in human and in mouse. So transcription factor binding, DNA accessibility, and gene expression across hundreds of cell types. And it does so not at single nucleotide resolution, but in bins of 128 base pairs. So kind of the summed expression, the cage sex signal in a certain bin. And this model is very successful if you ask it to predict gene expression of genes it has not seen in training, and which points to it having learned definitely features that explain how genes are expressed in the cell. However, one difficulty we have, of course, is this is a huge model, and it's not very clear how it actually manages to make its predictions, how it manages to correctly predict the expression of a gene it has never seen. And this is a well-known problem in the field, of course, that these deep models are black boxes. We give them sequence, then they do something with it. Somehow there's a lot of matrix multiplications, non-linearities, and so on. And at the end, it gives us a prediction, but we can't easily point out what features it has used, what kind of principles. But this being said, this problem is not new to biology, because in a sense, the cell is equally a black box in that way, because what experiments can do, we can put DNA into the cell, then things happen inside of the cell, and then we can have some kind of experimental measurement. But generally, the cell also won't tell us how it has arrived at producing this experimental measurement from the DNA. So what actually happens in the cell is also to some extent a black box. And this is fortunately not such a big problem, because biologists have figured out very smart ways of studying this black box, which is the cell. And most importantly for us today will be so-called perturbation experiments, where either DNA is inserted into the cell in form of a plasmid or some kind of reporter assay, or with a virus, or where some CRISPR system is used to edit the DNA. And then we can see when we do this perturbation, what happens to the experimental readouts we get in the end. And in this way, one can try to find the principles by which the black box works. And of course, the same approach, I mean, this is a very basic idea. I'm telling you, this is the controlled experiment, essentially, which I'm sort of reinventing in a way, reinventing the wheel for deep learning models, because there, of course, you can do exactly the same thing. You can also edit the sequence, and you can see how does the predicted expression change. Obviously, none of this here is particularly revolutionary, but my idea was to just take this idea and run with it, as they say, and basically take all of these, or not all, but a significant fraction of these recent, very well-designed reporter essays, targeted perturbation experiments, and see what happens if I sort of rerun them in this Enformer model. Will it correctly predict what these perturbations do? And what does this tell us about how the model thinks the regulatory code works in a way? I will focus primarily on 3 questions related to sort of 3 features of the regulatory code. First, I will tell you a bit about proximal regulation. So how do small regulatory elements close to the TSS of the gene they control, how do those drive expression? And how do individual elements in this small sequence determine its regulatory importance? Next, I will talk a bit about distal regulation. So this is the special feature of higher eukaryotes, right? That we have these regulatory elements which are very far in the linear sequence, but somehow manage to nevertheless interact with the promoter and affect expression over a long linear distance. And finally, I will talk a bit about the relative what the data tells us about the relative importance of these two mechanisms. So first of all, about this proximal regulation. Now, this is, from what I can see from the literature, this is probably the most extensively studied component of regulation. And this is to a large extent because the current techniques we have lend themselves very well to study it. And there are very many technologies nowadays, such as so-called massively parallel reporter assays, virus-based reporter assays, StarSec, there's the Sure technology, and others. And all of these technologies ultimately rely on having some sort of reporter gene. The transcription of this reporter gene is then controlled by a cis-regulatory element, which we design or which we take from the genome, or which in some cases is entirely random. And it's inserted right before this imported gene. And then this whole construct is either put in the cell as a plasmid or integrated into the genome. And essentially then this small regulatory elements acts as a sort of promoter. It may have some motifs or so on, and we can measure how these different elements then drive the transcription of this reporter gene. Now, there are a lot of these experiments, but for most of them, at least for every single one I tried, and I tried several, Enformer will tell you, will usually predict you very well what the regulatory strength of such a small promoter or CRE fragment will be in such an experiment. Here I show one example study from Jesse Engritz's lab, where they tested 1,000 promoter fragments. And as you can see, the model can predict you very well what the expression of such a promoter fragment would be. And I want to note here that for this and for everything else I will show you today, I've never fine-tuned Enformer. I take the model as is. It has never seen this particular data. It has seen these promoters because they come from the genome, but it has never seen them sort of individually tested alone. It has always seen them as part of the genome. So it can correctly predict what they will do alone to a big extent. It can do even more than this. There's an experiment by the lab of Aaron Segal. And what they did was they had a fixed promoter fragment, and then they inserted or removed individual transcription factor binding motifs. And then they measured what is the impact of individual motifs if we put them in or remove them. And as you can see, there's obviously some noise. Part of this is because the assay itself will be noisy. Part of it will also be model error. But generally, the model is quite good at predicting how does one small change in the motif, so one motif more or less, how does it change the expression driven by this fragment? And I would say for a model which looks at so much of sequence and which has never been trained on such tiny perturbations, it's actually quite impressive that it gets this very right. And here, this is my favorite kind of visual example. The same lab as before also did an experiment where they take a Tata box and they shift its position in the sequence. They create kind of a fragment where every time the Tata box is shifted a bit, and they measure again for each one. And you can see there's sort of an offset in the prediction, but the trend is predicted very well by Enformer. So the measured values and the predicted values track each other very well. So this very subtle shift of the Tata box, the model can tell you quite well what it will change to expression. And now, particularly interesting, we see there's this huge drop in expression if the Tata box is moved to this -50 in this particular background where it was tested. And the experiment itself doesn't tell us whether this is because the Tata box doesn't like this position or whether it is because something else is disrupted. But the model will actually tell you its view of the world, so to speak, because we can check what happens if we introduce some other perturbation there, which is not the Tata box. And actually, we can see that if we mutate individual bases, we already have a strong effect. And this is because at this -50 position, there's actually the motif of another TF. It's a cat box motif. And so what's actually happening here is that the Tata is disrupting this other motif, and it has nothing to do with the position. And the model tells us this. So in a way, you can even use it to interpret experimental results. This being said, it is not so that Enformer has learned sort of everything. So obviously there's some quantitative error, as we saw in the plots before. It's not a perfect predictor quantitatively, but there's also some qualitative things qualitative flaws we can find. One example is in this field, which is now, there's a lot of interest in this, which is the possibility of, is there a grammar between TF motifs? So there's not just a question, how do individual TF motifs drive expression, but also, is there certain rules of how motifs are combined to change their meaning? And there was a recent study, very famous by now, I guess, where using single nucleotide resolution binding data, they discovered that there's a transcription factor called NANOC, which if it's placed at if two motifs are placed at a certain distance, then they cooperate and they bind stronger. So you see this in this plot. Essentially here you have a motif at 0, and then we insert a second motif at a certain distance. This is a prediction from a model, but it's experimentally confirmed. And we can see that at a distance of 10, the two motifs cooperate to increase binding. This is actually because the DNA has a turn, and at this distance of around 10 base pairs, the two motifs would be aligned on the double helix, so they can actually cooperate. Now, of course, we can run the same kind of analysis in Enformer, and we can ask whether we see a similar pattern. So I looked at the binding track for this motif, and I did the same kind of motif spacing experiment. As you can see, well, you can see nothing. Nothing happens. So this kind of subtle pattern, it seems to not have learned at all. And it doesn't care if there's a second motif. It has no change to the binding. This being said, I don't want to say that it never learns this kind of cooperation between motifs. And there's another example from the same paper comes regarding these two factors. And there, you could say that the pattern that Enformer will predict you in terms of the effect of binding of, I think, two motifs is at least qualitatively similar to the pattern predicted in this BPNet paper. So there might be some interactions, but to learn really subtle ones, such as this NANOC, it is possible that we need this single nucleotide resolution model. Okay. So now I've told you very many things about this highly local regulation, but of course, Enformer is this huge model. It sees a huge amount of sequence. So maybe it's strange to ask it to predict what happens if I move one motif a few base pairs. And rather I should ask, like, does it get this distal regulation? And this, of course, is a very important topic because one of the kind of biology 101 ideas of deep models, of eukaryotic regulation, right, is that there's this interaction between elements which are far away in the sequence. So the promoter is not the one running the show necessarily, but rather there's all these far away elements which cooperate to determine expressions. Of course, this is a lot harder to study than the proximal regulation because it's not necessarily possible to do an MPRA with hundreds of kilobases. Maybe there's other ways. But as a result, we also have less data on this. And to my understanding, our general understanding of this is quite more limited. However, there were two recent targeted essays. They both have their limitations, but I think they're quite interesting to note in this regard, because to me, they define a sort of null model or a kind of baseline model of how distal regulation could work. The first of this recent papers is actually a plasmid-based assay, where you have a lot of promoter fragments, you have a lot of enhancer fragments, and then you test every possible combination. And now, obviously, because this is a plasmid-based assay, one can make very many criticisms of whether this truly captures promoter-enhancer biology, but it is somewhat, it at least gives us some insight on how these elements interact. And interestingly, what the authors found was that the interaction between these elements is actually very simple. So basically, the expression driven by the combination of a particular promoter with a particular enhancer is very well approximated by just taking kind of the average effect of the promoter and the data, the average effect of the enhancer, and multiplying these two together. So there seems to be no like kind of combinatorial rule that enhancer A only works with promoter B or something like this, but rather it seems that each one has their intrinsic strength, and the combined effect is just the multiplication of the individual ones. This is the first part of my null model, that all the interactions are multiplicative. And the second part of the null model comes from another recent paper by the lab of Luca Giorgetti. And there, what they did is they had they took an unstructured region of DNA and they ensured that it's unstructured by removing the CTCF binding sites and all this stuff. And then they added a promoter, they added an enhancer, and then they used the transposon system to shift the position of the enhancer relative to the promoter. And for each shift, they then measure the expression, which is driven by this. And they then fit a very subtle and complex biophysical model. But my very reductive explanation of their result would be that essentially the effect of the enhancer scales more or less proportionally to distance. So it decays with distance, and it decays proportional to 1 over distance. So it's like a hyperbolic decay. And if you put these two things together, then my baseline or null model of distal regulation essentially is that the expression, if you have considered just one enhancer, is driven by the strength of the promoter times the strength of the enhancer, which is corrected by dividing through with the distance. And it's noteworthy that this is essentially equivalent to the activity by contact model, if that's something you know, because the contact is actually quite well approximated by the distance. And now I can ask, well, does Enformer agree with this null model of distal regulation? And for this, first of all, I did this sort of enhancer-promoter combination assay to see if the interactions are multiplicative. And again, I took a lot of promoters, took a lot of enhancers, tested all the combinations, see what Enformer predicts for all combinations, and then see what a multiplicative model based on the averages would predict. And as you can see, the fit is almost perfect. So it seems that in principle, Enformer agrees very much with this multiplicative idea. And secondly, we can also do in Enformer this enhancer shift. So we can just take promoters, take enhancers, and shift the position of the enhancer relative to the promoter. And I measure then, kind of to measure the effect of the enhancer, I measure the change in expression if you kind of shuffle the enhancer. So you destroy the enhancer, and then you see how much does predicted expression at the promoter change. And you can see that this is decreasing in distance, and it's a sort of exponential decay. So this is a log-log plot, and if you go down, but if you increase the distance by a factor of 10, you lose a factor of 10 effect. So in this sense, the Enformer agrees quite well with the null model. And now the question is, does this null model also describe what we know about distal regulation in the genome? And for this, I looked at these CRISPR interference studies, which try to find the effect of enhancers in the actual genomic context, so without the targeted assay. And what they do is they have a CRISPR system which delivers a repressor to an enhancer. And then if this enhancer actually cooperates with the promoter, it will deliver the repressor to the promoter and thus affect expression at the promoter. And what I've plotted here in red is for two of these, the two biggest known of these experiments, what was the measured change in expression for enhancers which in these assays had a significant effect. So I don't look at the negative set, I look just at the positive set, and I see, is there a relationship with distance? And we see for these positive enhancers, there's in the assays, there seems to be no relationship with distance. So the strength is not related to distance. It's important here to note that this doesn't actually contradict the previous thing I showed you, because the previous thing where the enhancers moved, it's always the same enhancer. And here we are comparing different enhancers. So the fact that there is no decay here doesn't mean that there isn't this decay at all. But what it means is that these far away enhancers somehow seem to escape the null model. And whereas what we see with Enformer is that Enformer kind of more or less religiously follows the null model. There are exceptions. There are some distal enhancers where Enformer does predict a strong effect, but generally it will decay the effect of further away enhancers quite rapidly, and similar to this 1/distance factor. So as I said, what seems to happen is that somehow some enhancers in the genome seem to escape the null model. And of course, we don't just know this from this data. There's some famous example from previous experiments. There's, I think the most famous is the SHH enhancer in mice, which is 1 million bases from its target gene. But we know for a fact that if you mutate it, it affects its target gene. And as a result, like baby mice will not have long legs, but they will have very tiny legs. So obviously this enhancer is doing something very important. And it seems to do this by escaping the null model cell. Now the question is, how do they do it? Obviously the null model has two components, or there's two things that could be overcome, or a combination of both. One thing that could be is that potentially they overcome multiplicativity, so the fact that everything is just a multiplication. This could be overcome in the genome by some kind of cofactor which is required for distal enhancers, but which isn't relevant in the plasmid context that Bergman et al. Tested. My interpretation of the possibility here. The second thing where the problem could be is that there might be or it seems quite likely that there is some kind of folding code in the genome that gives the genome a 3D structure, which then allows far away enhancers to associate with certain promoters. And it could be that the model is actually missing this. If the latter is the case, by the way, then this could also mean that potentially what you need is an even much bigger receptive field, because this kind of folding code doesn't need to be in the enhancer sequence. It doesn't need to be in the promoter sequence. It could be anywhere in between, right? Or somewhere entirely different, which of course makes the task quite a lot harder. Now, coming to the third part, speaking about this large receptive field, what I showed you now is that this model, which has this large receptive field, is actually really, really good, in my opinion, at understanding proximal regulation. And even though it's certainly better than many alternatives at understanding distal regulation, it seems to overall, in an absolute sense, not be particularly good at it. And this now raises the question, why is it good at predicting gene expression in general? So it seems to be that, you know, it can integrate longer information and it can predict gene expression quite well in general, but somehow it isn't using the distal information. And we can actually make this sort of concrete, this idea, by testing what happens if we predict gene expression, but we give the model less sequence to work with. So instead of giving the model its full receptive field, we just throw away the distal part of the sequence, mask it out, and ask the model to nevertheless. And we did this, and we asked like how much of, say, gene expression variation can we predict in GTEx or another big RNA-seq study of human tissues. We look at both between condition and between gene variation. And in all cases, we see that already with a very small amount of sequence, so just with 1 kilobase around the TSS. So this is 0.5% of the actual sequence the model could access. We already do quite a lot. So we do more than half of the total performance we can achieve with the model, just with this tiny amount of sequence. And then as we increase the amount of sequence, we see that we quickly run into diminishing returns. So with 1/5 of the sequence, we already do almost the same as we do with the full sequence. And this is true both for between gene and also for between condition variation of gene expression. I would have expected that here it's more stark, but actually it seems quite similar. And this, I want to stress here, this is not entirely wrong or entirely surprising either, because actually we know that most known regulators seem to actually be close to the gene they regulate. And that's also maybe not so surprising. But here I've just made this explicit in a plot. It shows the distribution of known regulatory elements. How do I impute known regulatory elements? I look at kind of the validated enhancers in this previous assays. I also look at known EQTLs. And I ask how far are they from the gene they regulate. And this is, by the way, limited to the Enformer receptive field. So obviously there could be stuff further away. I don't include this right now. But what we see is that within this receptive field, most of those things which are actual regulators will be relatively close to the gene. And actually, there's a recent big MPRA study done by Vikram, and they show also that with very small fragments, and you test them in an MPRA context, you can also predict very well the endogenous gene expression of the whole in the genome, of the gene in the genome. And so it does seem the case that it's true that the proximal sequence is doing most of the work, at least in a genome-wide sense. What, however, makes the task of learning distal regulation even more difficult now is that if we ask, where are the things which could actually be enhancers, then we see that these, and I should say how I proxy these I proxy by looking at this so-called ENCODE screen study, where they sort of defined what are biochemical marks that could designate an enhancer. And I take the position of all of these. And what we see is that you can find those basically at any distance to a gene. So they are sort of uniformly distributed. They are not bunched up close to actual TSS sites, but they are sort of uniformly distributed on the genome, which means that as you get further from the gene, the task of discriminating becomes more and more hard, because you will know that most of the stuff won't matter. But there's still as many things which look like stuff that could matter, at least in terms of these biochemical marks. And so as a result, it's probably quite hard for a model to learn this distal regulation, because it seems to quantitatively do, at least in a genome-wide sense, not so much. But there's a lot of candidate sequences which you could select to say that they are important, but only few of them will be important. So it's a task with a lot of noise and very little signal in some sense. And the signal sort of gets worse as you go further. So that already brings me to the end of what I wanted to tell you here today. So the main takeaways would be that currently most of the signal that this model seems to be learning comes actually from the proximal sequence. And again, this is not completely unreasonable. This is maybe where a lot of the signal is. And the models learn this signal very well. They generalize quite well to very small perturbations, which they have never seen in training, at least not in this way. And there might be some kind of subtle relationships like this motive spacing grammar, which are only accessible if you learn in single nucleotide resolution, which Enformer didn't do. So maybe it misses some of those, but a lot of the proximal regulation it definitely gets. Then in the distal regulation, it seems that what the model has learned is mostly this null model, what I call the null model. And this is again not unreasonable. It matches recent assays, as I showed you. But clearly, there are cases in the genome where this doesn't hold. There are enhancers which somehow overcome this. And to crack this may require different data, because as we can see that in the current data, the signal coming from these distal elements seems to be quite small. And there might be very complicated processes that are actually driving this. And so the signal to noise ratio is just quite a challenge to overcome. Yeah, that would be it from me. Thank you.